Hi, everyone. Good evening. Welcome to tonight's info session. Today, the KRTP, alongside Brad Hogg, That Bee Man YouTube channel, and the Canadian Beekeepers blog YouTube channel, is hosting Agrogene Solutions. So we have our guest speaker, Pascal Michaud, and she's going to give us an info session on what Agrogene Solutions is all about in terms of hive diagnostics. So I'm just going to introduce Pascal, and we're going to get the presentation started. This will go on for probably about 15 to 20 minutes, and then we'll take questions. So as questions come to you, you can write them in the chat of the YouTube where you're viewing this. All of those questions will be fed directly to our online studio, and we'll be able to pose them to Pascal at the end. So please put your questions into the chat. So Pascal Michaud is the co-founder and CEO of Agrogene Solutions, a biotech startup focusing on honeybee health, monitoring, and diagnostics. With a doctorate in molecular and cellular biology from Université Laval, Pascal brings a scientific background in biochemistry and gene regulation. So after returning to New Brunswick in 2020, she joined the University of Moncton as a research assistant, where her work laid the foundation for the launch of Agrogene Solutions. So with that, Pascal, I'll leave the screen and you can bring up your presentation and get started.
Perfect. Thank you for the introduction, Matthew. All right. Yeah, so in the next 15 to 20 minutes, I'll just tell you a bit about Agrogene Solutions, what we do, and what hopefully we can do to help you and what we can offer. So I'm sure everyone here already knows what the problem is. We are losing a lot of hives. We were just discussing the numbers of overwinter losses last year. It's very variable. Some provinces were hit harder than other, but it's really a worldwide challenge. One of the main reasons is because we are seeing increasing hive infections, and therefore less bee populations. And then what that means is obviously what's being hard to sustain as sustainable beekeeping for you. So we really believe that the solution lies in a preventative approach to disease management. So what we offer is to detect infections earlier. So the earlier you detect an infection, the better chance you have to do something about it. We also offer to evaluate hive health. So we can look at the bees' overall health. We can measure their immune response and their energy levels to really get a global view of what's happening in your hive. And then we can also monitor resilience and trends over time. All of this to help you make better hive decision management. So how does it work? So you can go directly on our website and choose and order a test. We offer 3 different types of tests, which I will explain in the next slide. So this can be bought directly online. However, I will say that we are working currently on getting these tests in the stores. So keep an eye out. We've been chatting with a store in Manitoba. So keep an eye out for that to go buy the test there. But in the meantime, you can definitely just order directly on our website. Once you order a test, we're going to send you some sampling material. So it's a quick and easy sampling for the pathogen detection. We only require a swab. So you just have to swab the hive entrance to get the genetic material from the bottom board. And then depending on the type of test that you purchase, we also require a couple bees, but 3 to 5 to around 5 to 10 bees is usually what we ask for. And then so you get your sample, you ship them back to our lab. So we make it easy. There's a prepaid label in there. So you just have to drop it off in the mail and send it to our lab. And then we will analyze that and give you back a hive report. And we aim to do that within 3 days of us receiving the sample in our lab. So we really try to be quick in the way that we deliver the results. So as I mentioned, we offer three different type of testing. So the first one is just looking at pathogen detection. So we will detect and quantify a panel of 10 pathogens. So we look at different viruses. We look at Nosema, EFB, AFB, and Varroa as well. And we kind of came up with this little color-coded system just for it to be easy to view and understand what's going on. So green would mean that we have not detected this particular pathogen in your hive at all. Yellow would mean that we detected it, but at low levels, not too concerning. Red would mean that now it's detected at high levels. And then the black one is really, I would say, extra high levels. We usually only see the black detection when it's a hive that is already lost, and we can kind of see what happened. But usually in hives that are still alive, we will range between the green, the yellow, and the red. So that's our first test that we offer, just the pathogen detection. The second one is a bit more complete. So this one we call the overall health assessment. So you still get your panel of 10 pathogens, so the same ones. But we also look here at immune system activation and the energy reserves. So you can also get a sense of really what's happening with the bees. Are they in good health? Are they able to fight these infections or not? And then the last one is more of so we call this a seasonal monitoring. So it would be the same test as the overall health assessment, but then you get 3 tests throughout the season. And then we really recommend doing this at key time points, being in the spring, in the summer, and in the fall as well. And I will touch back on this at the end just to give you a bit more of an overview of what type of results and what type of insights you can gain from using the seasonal monitoring. So really what differentiates us from other lab services is that we are the first complete and preventative approach to disease management. So we're a team of scientists. So all of this obviously is science backed. We systematically measure all pathogens. So you don't have to tell us what you think is wrong with your hive. We just look at everything because you never know what could be in there. So every test, we include those 10 pathogens. We're always on the lookout for new ones as well. So unfortunately, new ones will keep appearing, but we will just add them in our testing as they come. Also offer non-invasive sampling, so a couple bees and just a swab of the entrance just to make it as quick and easy as possible for you to sample. We deliver results, as I mentioned, quickly. So within 3 days of us getting the sample in our labs, you will get your report back. And we're also the only ones who look at this hive health analysis. So these specific biomarkers and these genes that we look at in the bees directly to give you that overview of the immune system and the energy reserves. So who's behind this? As I mentioned, we are a team of scientists. We're all based at the University of Moncton in New Brunswick. We have different backgrounds that really complement together. So ranging from molecular biology to immunity to insect metabolism and to genetics. So we really combined our expertise to bring this to beekeepers, to have a tool that's easy to use and that gives clear results. So we started this project because we ourselves maintain hives at the University of Moncton. So we usually maintain between 3 to 5 hives. We actually keep them on top of a building directly on campus. And we actually have what we call these smart hives. So it's a collaboration with a company called Hive. And we have these sensors on the hive that measure weight, humidity, and then the interior exterior temperature. So that's really great for all our ongoing research projects. And really, we started the company because we ourselves were losing hives due to infections, and we just wanted a quicker and easier way to detect them. For the last few minutes, and I've told you a bit more about us and about what we do, I just want to circle back to that seasonal monitoring that I had mentioned. And I just want to show 2 case studies of what type of results we see and what they mean. So these come from a big study that we did a couple of years back. So we analyzed over 200 hive samples from New Brunswick and also from the East Coast of the United States. And then we followed them throughout the season and really tried to see what were the insights we could get from that and what were the patterns that could predict good hive survival. So I'll show you a first example here of seasonal monitoring. So for a particular hive here, we can see in the spring. So quickly, if you look at the pathogens up top and you see the colors, you see there are some. This was the summer where we had a lot of EFB. So the EFB was here in red, a couple other things popping in yellow in the beginning of the season in the spring. But the immune system was already kind of activated in these hives. So then we sampled a bit later in the summer. Again, we see that we have some infections. So here there was a particular virus that was popping up as red, other things in yellow. But now the immune system had fully kicked in these hives. So then if we look in the fall, you look at the infections here, the colors, so a lot less infection here. So the hive kind of had naturally fought off the infections, and then the immune system was brought back down to a lower level as this hive entered the fall with a low level of pathogens. And then we also followed the same hives the next spring, so the following years. And so this hive had survived the winter. And this is one example, but we saw many of these and really kind of showed us that the hives that respond quickly to infections in the beginning of the summer, meaning that their immune system was activated, was a good prognostic for these hives. So these were the ones that were usually going to make it through the winter. Then the other example, obviously I'll show you the opposite. So we had a few hives where we kind of saw different patterns. So this one in the spring, not a lot of infections. The immune system was not activated. But then came summer, and now we were seeing quite a bit of infection. So 2 viruses here in red, EFB in red as well. So that's already kind of a red flag. And then on top of that, as you can see here in the summer, the immune system was not kicking in. So it wasn't getting activated, try to fight off these infections, which then led us into the fall. So again, even more infections here. The immune system here kind of started to kick in the fall, but it was a bit late and it didn't fully kick in here. So this hive went into the winter with a lot of infections that were not being fought off by the bees. And then I would like to show you the report from the next year, but unfortunately I cannot because that's one of the hive that was lost over winter. And again, this is one example, but we've seen quite a bit of these signatures. So in this case, what we conclude from that is that the hives that don't respond to infections early in the summer, meaning if their immune system is not activated, not a good prognostic for these hives. So that's where, they would probably need more help, more treatment. And so that's really the advantages of using the seasonal monitoring is you can see things progress throughout the season and, decide when to intervene if you see some infections that are, getting a bit out of control. So yeah, that's it for me. So kept it pretty quick, but I am happy to keep some time here to answer any questions that anyone might have.
Hi, Pascal. Thank you. So you can hear me all right?
Yeah.
Yeah. I guess firstly, let's say I order this kit and I get it in the mail. So if we think of a typical operation with about 40 colonies in the yard, and a lot of us are looking at our colony health varroa levels as such on a basis of bee yards, And we know that in different areas, we might have higher varroa levels or lower and stuff like that.
Mhm.
So if I grab a swab, and am I going out there and am I swabbing 5 hives? Or am I swabbing 10 with one swab or 20?
Yeah.
Yeah, and secondly, I guess, what, like, how, can you just walk us through the process of getting that swab, and actually, is it on the bottom board? Is it on the frame? Debris issues that might maybe impact both detection, but also in terms of your capacity to actually process it.
Yeah, it's a great question. So first of all, for your first question, we would recommend just swabbing per yard, especially if you have a large operation. You're not going to go and sample every single one of your hives necessarily. So if you want to just look at a yard in general, you can definitely, we've had beekeepers swab a few bottom boards within the same yard. As you mentioned, you will kind of see the same patterns in those hives anyways, in terms of varroa and in terms of everything else. So it's definitely something that can be done. If you want to look at a yard more specifically, you can swab multiple bottom boards in that yard. And then for your second question, so I've had swabs come in with a bunch of debris in it, and I was still get good resolution. So it's a PCR based approach. So we really will single out the DNA of the specific virus or bacteria or whatever we're looking at. So usually they will just swab the bottom board. If we've had people just swab the frames as well, if they prefer doing that. What we want to capture is the environmental DNA, is what we call. So just an overview of what's in the hive, and just that environmental DNA is sufficient for us to detect and quantify in our test.
So it doesn't make a big difference in terms of in terms of the science and detection, whether it is the bottom board or the side of a frame.
No, and we tested it because some beekeepers had some bottom boards that were like a screen. And then in that case, it was better to swab the frame. We tested it as well, but we see the same results.
Okay, before I ask any more questions, I'll just go through a few in the chat here. Firstly, I'll just pick on an easy one. It's a question and comment that just in terms of the reporting, I'm not sure how you chose the colors for each one, but just keeping in mind colorblind beekeepers who might not be able to tell the difference. So something to keep in mind, or maybe you've already looked at that. But.
Yeah, no, that's a great comment. And then we're always available. It's not like I send a report and then never speak to you again. Like, we're always available if people have questions and, want to understand a bit more what it means. We'll do the best we can as well. So if anything is unclear, we're definitely always there to help.
Okay. Another easy one here before we move more back to the kind of bee questions. Is this available in the US? I mean, is there an issue with mailing that across that type of stuff across the border?
Yeah, that's a good question. So we did have some US collaborators a few years ago who sent us some sampling. We have to figure out some shipping issues before we can offer it to the US, but it is in the plans to offer it soon. So keep an eye out for that. But there is a little bit of shipping border control that we just need to figure out. So right now we're offering to all of Canada, but it's definitely our goal to bring it to the US sooner than later.
Okay, now I'm going to come back to the bee sort of oriented questions. Jason Seward says, if their immune system doesn't activate, I feel the colony should be requeened. Any comments on no activation of the immune system? And also maybe you could talk a little bit about, and maybe you went over this in the presentation there, but like, what does that all mean by how do you determine how well the immune system's been activated?
Yeah. So that's a good question. I know I've had some beekeepers who would requeen if they were seeing results where they had some high levels of pathogens or whatnot. So we kind of want to get to a point where we can, give better recommendations, as if you get this exact report from us, this is what you should do, For now, we still want to work with beekeepers and, get their feedback, see what's worked best for them. We're also working with our inspectors to really, get their feedback of what they would be the best recommendation on what to do for that. But how we measure it really is we look at some genes that we know from our research that we did at the university, and they're genes we know correlate with the activation of the immune system. So it's not just one gene. We look at a subset of specific genes. Again, that came from our research in the previous years, and that's kind of how we came with this just simple 3-step activation of low, medium, or highly activated. It's really a subset of biomarkers that we know represent the immune function.
Sorry, so maybe I just missed a bit. So you actually have to sample the bees and then look at the bees?
For that one, yes. So if you just do the pathogen test, we only require a swab. If you want to get the full test with the pathogen and the health analysis, we do need a couple bees as well for that sample.
Okay, that makes sense. And that kind of leads into the next question, which kind of goes a step back here, but I think it's really important when we're following these protocols, and we're going to get this package in the mail from you. So I've ordered the package. So a beekeeper receives it, they open it up. What do they see? And what do they have to do exactly? Yeah. So the question was, can you detail exactly what I can get, what I get in the test kit, and exactly the protocol on how I take the sample?
Yeah, absolutely. So you'll receive an envelope in there, depending on the test you chose. So if you have just the pathogen test, you'll have a swab. If you have the overall immune health and pathogen, you'll have a swab, and you'll have a little plastic container to sample the bees as well in there. Every test also comes with the instructions in the envelope you'll receive. So it's detailed in there how to insert the swab, and then rotate on the bottom board, how many bees you need. So we have all that detailed in the instructions in there. And then you'll also get a return envelope with a prepaid label to ship it back to our lab. So shipping is always included in our prices as well. So you don't have to go and pay for shipping to send it back to our lab. There's a prepaid label in there. You just drop it off, and that gets sent to our lab. So we aim to make it as easy as possible.
Okay, so if we're sending the swabs back, I expect they're dry, so they don't need any special treatment. They just go back in the tube and get mailed out. And so for a bee sample, do you have recommended protocols on shipping? Should it be shipped dry on a Monday? Does it need to be dampened in ethanol?
Yeah, we ask that people keep it in a cool place. If they're gonna sample and then only ship it a few days later, we ask that they keep it in a cold, and so a fridge or something before they ship it. But then once it's in the mail, especially now since we're just working with Canada, the time it takes even to get from coast to coast, we've tested it and left the samples out at the room temperature for that long, and that transit time doesn't affect even for the bees as well.
Oh, great. So it's very simple to mail these off. The next question from DCBs was, wouldn't a hive that produces higher levels of propolis in turn have lower immune activation?
That is a great question. I'm afraid I don't have the answer to that one. Yeah, I can't speak. I can't say for sure on that one.
Okay. The next question from a Canadian Beekeepers blog: how are you able to determine pathogen levels by swabbing the bottom board? I'd assume you need to test the actual bees. So I guess the assumption there is that you swab, and yes, you can know present or absence. But how can you explain a little bit about the science behind that and how you can actually tell us the level?
Absolutely. Yeah. So I'll start by saying that when we tested this, we would get a sample of the swab and then a sample of the bees, and then run them both parallel and see if we had the same results. And we did. The reason why we can detect it, as I was mentioning, is what we call environmental DNA. It's the same way that, you can take some wastewater, and just from the environmental DNA in there, you can kind of track what fishes were around and all that. So it's kind of the same concept applied here, literally. So whatever DNA is released by these pathogens in the environment is sufficient with the PCR techniques that we have now. It's sufficient to be detected by just a swab. So we tested both when we started, and then when we realized that the swab replicated the same results we were seeing in the bees, we were like, well, we're going to take the easier route here, and the swab's definitely easier and gives the same result, really just by the environmental DNA that's released by these pathogens in the hive.
I guess one of the things that really interests me we've heard a little bit here in Western Canada about a European foul brood popping up a number of times. And I mean, I'm no expert on it, but just on observations over the past few years, it seems like there's noticeable by the beekeepers in advance that something's not really right, and then they see their first case, or they're made aware of their first case because they haven't seen it around for 20 years or 30 years. And at that point, it is, it's you have one severe case in a yard, and then the rest of the bees in the yard do have it at low levels. So I guess I'm very interested in trying to utilize this tool and work with it to detect low levels of something like EFB, specifically EFB.
Yeah, absolutely.
In advance. Have you done any specific work there in the Maritimes with EFB, or like, I mean, I know you have tested it and if you've done the trials, but just wondering if you have any further comment on extensive work with EFB.
We haven't been seeing a lot of it in the last 2 years. We saw it more 3 summers ago, and then the summer after that we were seeing a lot of Nosema. And then personally for us this summer and last summer, the biggest problems were viruses. So we had the deformed wing virus that we were seeing a lot of at levels that I had never seen before. So I can speak for us because that's, the most samples I've received was from the East Coast, and that was definitely the issue that we have been seeing here in the last few years. Haven't been seeing as much EFB right now, but again, I can only speak to the samples that I have analyzed here in the Maritimes.
Okay, and so that just kind of brings up a question on Nosema I had, and apologize if I missed this. So for like a low, medium, or high level of Nosema, what's your thresholds?
So it depends on, so since it's a PCR test, our thresholds are based on what we call the CT or the CQ value. So how many cycles it takes for it to be detected. So the way we did the color coding tests was with that big trial run that we did with 200 samples, and the physical signs that the beekeepers were seeing, and then from there we kind of determined what was background noise, what was more higher level, what was more concerning. But we do have a project coming up with our tech transfer team here in Atlantic Canada, where we really want to correlate it with the spore count as well. So for now, I can't tell you exactly like a CQ of this much means that many spores, but that's something that we plan on doing really soon.
Oh, great. We'll be looking forward to that. Just a comment and question here. A Canadian Beekeepers blog says, when I used to send samples for virus, they were fussy about the freshness of the samples. Does prolonged time deteriorate the result?
Not in our case, not with the swabs. Again, I've tested it. I left it at room temperature for a week, and it didn't affect in our case because the swab is dry. I even tested to see if it was better to put it in some liquid, and it's actually not. It makes it worse if you put it in liquid. So we found that just keeping it dry was the best way to preserve it, and we haven't had any issues.
So have you, or do you have any, do you have now the ability, or do you intend to have the ability to detect Tropolalops in the future?
We are talking about it, definitely. And again, that's something else we were discussing with our tech transfer team here. We're definitely on the lookout for it if and when it does become problematic. It's quite easy for us to just add a new pathogen in the test. We'll just have to do obviously a bit of validation first, but we are on the lookouts for anything new that might arise, and that's one of them.
So potentially you would be able to add that as an option long before we hopefully never see the light of day of tropical relapse, but you're able to add that to your
It's one of the research projects that we have going on pretty soon as well.
Great. I'll make a last call for questions, and in that time, if any of our hosts or if you think of anything else you'd like to add, then please go ahead. I guess one of the things could be interesting too, I don't know if you've talked about providing this test to other countries. I mean, currently we import package bees from Italy, New Zealand, Australia, queens from mainland US and Hawaii, and package bees from Chile as well. Have you had any connections? Because I mean, and that's fine if you haven't, but I think it could be interesting to down the line be able to incorporate some of this into some of our biosecurity practices.
No, of course, it's definitely one of the long-term goals, and it's a problem internationally, so it's not just a problem in Canada, unfortunately. So it's definitely one of the longer goals. For now, we're focusing on Canadian beekeepers just because of the shipping, and soon the US. But yeah.
Cool. I'll just take the final kind of questions and comments here, and we can wrap up. Brad from That Bee Man says, I like that you test for all pathogens. When I have a sick colony, I usually don't know why, and that's the reason I really need to know. Yeah, so just a final comment there, I guess. But if you have anything else you'd like to add, I'll let you. Otherwise,
no, I mean, we're always available. Like, we like to chat with beekeepers. If people have questions, you can reach us at info@agrogenesolutions.com, or on our website. There's also a contact us box. So if anyone has any further questions or just wants to chat, I'm happy to answer later questions.
Perfect. Okay, well, with that, we'll end today's session, and I appreciate your time. Thank you very much. All right, good night, everyone.